Today I'll talk about my research project and the development we've done so far. I'm working on the development of a topical delivery system consisting of lipid nanoparticles loaded with gene editing tools for the treatment of genetic skin disorders. Genetic skin disorders, also known as genodermatoses, are disorders caused by genetic factors with skin manifestations. These manifestations, they can go from impairment on the skin permeability, affecting the transport and the retention of water and nutrients in the body, severe inflammation reactions, and even life-threatening conditions. But despite the severity, genodermatoses, there are little or no treatment available currently. The treatment options, they are only symptomatic, so the use of special baths and the constant moisturizing the skin, and there's no cure for them. The reason for this lack of treatment is because of the difficulty to reach the target site of the pathology. The skin layers, the epidermis layer is the most outer layer of the skin, and is usually the site of the cause of the genodermatosis. This layer is composed by the stratum corneum layer, which is the layer that we can usually see in our skin, and is a very thick and cornified layer that protects our body against external threats. And because of its composition, it is very hard to penetrate this layer. Many studies reported the use of lipid compounds as a formulation that is able to go through our skin. And here we propose the use of lipid nanoparticles, that is a nanocarrier formed by lipid compounds, and that stands out by the high bioavailability and the protection of the cargo. And once the lipid nanoparticles interacts with the cell, it is taken by endocytosis and can release the cargo in the cytoplasm. Since we're talking about genetic disorders, a possible way of treatment is through gene therapy. And here we propose the use of CRISPR in two different approaches. The first approach is with mRNA-based genome editing, where we load the lipid nanoparticle with mRNA for the Cas9 protein and the guide RNA. Then once delivered into the cell, the mRNA is translated to the Cas9 protein. And the second approach is the RNP-based genome editing, where we load the LNP with the Cas9 protein itself that is directly delivered to the cell. And as I mentioned earlier, it is very difficult to go through the stratum corneal layer in our skin. So a way to enhance this delivery, we are using a tool called microneedles, more specifically solid microneedles, that, as you can see in this image here, the yellowish layer represents the stratum corneal. Once the microneedle is applied in our skin, it creates openings in the stratum corneum, so then we can simply apply the formulation on top and they go through the inner layers. In this way, we can facilitate the topical delivery of the nanoparticles and reach the target site for the genodermatosis. Now I'll show some of the results we have. Initially, we wanted to figure out which LNP formulation would be the most ideal. So we did some screening through a few different formulations. Here we tested for the cell viability in primary keratinocytes treated with unloaded LNPs. We tested 3 different formulations in terms of compositions, A, B, and C, and 3 different concentrations of each component. So 1, 2, 3. And here we noticed that any of the formulations, they had significant decrease of cell viability in comparison with our control in primary keratinocytes, which suggests that the formulations were safe to use. Then we tested the same formulations for gene editing efficacy. The graph on the left is for the RNP approach that I mentioned earlier, and the graph on the right is the mRNA approach. And we can notice that all the formulations, they did induce editing on the cells, but only for the mRNA approach and the LNP-C, we noticed a significant increase of the gene editing efficacy compared with our positive control. Then we moved on to models that were more similar with the actual human skin. And here we can see excited human skin that we applied the microneedles to, as I mentioned, facilitate the penetration of the nanoparticles. The image on the left, we can clearly see the opening made by the microneedle use. And these red spots on the layer, they are usually stuck on the external corneal layer. This is the LNP. But with the opening created by the microneedles, we can see that the red fluorescence, the LNP, is going through the inner layers. And on the image on the left, on the right, it's an H&E staining. And again, we can see the opening and the disruption of the stratum corneum made by the use of the microneedle. And finally, we tested the same formulations for gene editing efficacy on excised human skin, that's the graft, the A graft on the left, and skin models, the graph on the right. We tested for both approaches, this time only using the LNP-C, which we found it was more effective. We tested for RNP and the mRNA approach. And we can notice that both of the approaches, they were effective for inducing gene editing compared with the control. And not only they were effective in leading to the gene editing, but also we can notice that both the skin, the human skin, and the reconstructed skin with the skin model had very similar results, suggesting that the skin model is also a reliable source for human research. So I would like to thank my supervisor, Dr. Sarah Hadrich, and all my lab for the help and support, the agencies for financial support, and of course, NMIN for having me. And just to finish, the key messages from my presentation are that the genodermatoses are genetic skin disorders with no long treatment that require more focused studies. The use of microneedles on the skin is an easy and effective method to improve drug delivery in the skin. And lipid nanoparticles are a powerful nanocarrier for the delivery of gene editing tools. Thank you for your attention, and I'm open for questions.
Great, thank you very much, Juliana. And I am pleased to open the floor to questions from the audience. You are all presently on mute, so to ask a question or join the discussion, you have to use your control panel to either raise your hand to speak or submit a written question. So Marshall, do you see any questions or hands at this time?
Presently, no questions or hands.
Great, well, that gives me the chance to ask a question. Juliana, you mentioned that it was difficult to deliver LNPs topically. Are you trying any other methods outside of microneedles to enhance the delivery?
Yes, actually, we are trying different methods. We are trying physical methods similar to the microneedles that is called microporation. We have recently received a laser device in the lab that we use to create pores in the skin, and that would have the same effect of the needles. And we're also trying to do other alternatives, like the use of cell-penetrating peptides that not only oscillate on the permeation through the layers of the skin, but also facilitates the transfection of the particle into the cell.
Great, thank you. And are there any plans for clinical tests in the future?
The whole project, it does involve one specific genodermatosis that's called ARCI, that's autosomal recessive congenital ichthyosis. It's a very harsh form of ichthyosis and is a monogenic disorder, so that's why we are targeting this disease. But the project is still in the early stage. My part was developing the concept of the proof of concept with the LNPs and the CRISPR. And then later on, we plan to move into disease models.
Great, thank you. And Marshall, to check in, any hands or questions?
Still handless and questionless.
Okay, I'll ask one more then. Juliana, could you speak to the skin model and how you made it?
Yeah, sure. I actually have an extra slide, if I can share the screen again.
Of course.
That would be more clear. Oh, here. In the lab, we have different protocols for skin models. The one that I use is collagen-based. So what we do is we try to replicate the skin layers in in vitro insert. So here we have the well that we put the media, and then we have an insert that goes on top. We mix the dermal matrix, which is the collagen gel, with fibroblasts. That's the main cells on the dermal layer of the skin. And then we create this gel with the fibroblasts. We put it on the bottom. We waited until this solidify, and then we just include the keratinocytes on top in a monolayer. And then as they grow, they're going to grow upwards since they are limited by the insert, and they're going to naturally create the different layers of the skin. So then we will have the dermal layer and the epidermal layer on top.
Excellent. Thank you. And Marsha, I just saw a question come in. Could you read that out loud for us, please?
But of course, this comes from Ariadne Tukmantel-Bido, who wrote, "Great talk. My understanding is that the genetic change is local. How long does the effect of the genetic change last on the cells, and how many reuses of the drug are necessary for the patient?"
Yes, this is actually something that we will have to touch later on the project. Right now, my main goal is delivering to the epidermis layer, but of course, the epidermis layer formed by different types of keratinocytes. And on the bottom of this layer is the basal keratinocytes, which would then grow into the different layers. Ideally, we are reaching this layer, the basal one, which would then lead to a more long-term effective. I cannot speak specifically how long would the treatment be. That'll depend a lot on the clinical test when we reach this phase. But at this point, I can tell that we are trying to develop methods to target the basal cells on the epidermis.